Hello, ladies and gentlemen. This is Tina from Mindray International Marketing and Sales Department. I have been working in Mandarin for about 10 years, 7 years in R&D department, and 3 years in the international marketing department as a clinical academic manager. I think it's a good opportunity to share the topic of Mandarin lab solution for COVID-19. I divided this topic into two parts, and the first part is the laboratory investigation for COVID-19, and the second one is what can Mandarin provide? Okay, let's come to the first part. Let's look at the pandemic of the COVID-19 worldwide, and this picture is from the WHO official website, and until February 2021, there are more than 100 million confirmed cases reported, and also more than 2 million confirmed deaths. The top five countries with the highest accumulated number of confirmed cases are the US, India, Brazil, Russia, and the UK. And the top 5 countries with the highest accumulative numbers of the deaths are the US, Brazil, Mexico, India, and the UK. The pandemic still seems far away from over. How can we overcome the pandemic? At the very beginning of the pandemic, we were worried about how to diagnose the new infectious disease and for what aims do doctors usually use different methods for the laboratory testing. And what are the clinical utilities of the serology test for the laboratory testing? And what is the real value of the serology test during the pandemic? As we all know that there are 4 common diagnostic tests for COVID-19, and they are molecular assays like nucleic acid test and or PCR, and antibody detection, antigen detection, and the virus isolation and capture. So among these 4 measures, PCR and virus capture are the gold standards, but virus isolation and capture need high requirements for capture conditions and have low sensitivity and long TAT. So it is usually used for research and not popular in the hospitals and the labs. And the advantage of antigen detection is the short TAT and the low cost, but it also has some disadvantages like high biosafety requirement, and there are also some difficulties of sampling which may lead to false negative. I would like to emphasize the molecular assays and antibody detection, and for PCR, it is also a good standard, a gold standard for diagnosis, and it has high specificity and can detect early infection of the disease, but it also need high requirements on the lab requirement, and there are also some difficulties of sampling and which may lead to false negative. The last one is the antibody detection, which I'd like to introduce more in the following slides. It has some advantages like short TAT, low cost, low biosafety requirement, high sensitivity and specificity, but it cannot be used as COVID-19 confirmation test. It can only support diagnosis. And this is the estimated course of the molecular and the serology biomarkers of COVID-19 patients. There are 2 effective ways for the doctors to detect the infection. One is PCR and the second is serology test. So at the very beginning of the infection, it is very hard to detect the virus directly, but around the time right before the symptom onset, it is a very good time point to do the virus isolation from the respiratory tract. And the medium shedding duration of the virus is about 20 days, sometimes could prolong to 37 days. So doctors usually use nasopharyngeal swab PCR to detect the virus directly during this period. Besides, doctors always prefer to use antibodies to support PCR to confirm the disease when the patients show up in the hospital with related symptoms more than 7 days. IgM antibody goes up at the time point, and it can exist in the blood for about 1 month. And it can indicate the acute infection around 7 days or to 10 days after symptom onset. IgG antibody increases, and it can exist for months and years, and it can indicate late and past infection. So What are the differences between IgM and IgG, IgG and total antibody? For the total antibody, it includes IgM, IgG, and IgA. It can indicate individuals' immune status, but it cannot indicate dynamic changes of antibodies and cannot help doctors figure out the disease courses. But IgM and IgG can help doctors to evaluate the dynamics of the immune response of the patients depending on the duration of the disease. Is the function of the viral test, the PCR, equal to that of serology test? And in the guidance for laboratory testing from WHO, serology tests can only support diagnosis in case where the nucleic acid tests are negative and the epidemiological evidence is strong. And the serology test can also be used for epidemiological assessment. And the confirmation test is the NAAT, such as PCR. As the epidemic gradually eased, more concerns followed. Do recovered COVID-19 patients have some protective immunity against the reinfection? Do antibodies confer reliable immunity? We now have some types of vaccines, although we don't have a specific medicine for COVID-19. And how can we know if we already have protective antibodies after vaccination? Is there herd immunity built after mass vaccination among the population? As the real situation of the herd immunity after mass vaccination is becoming the hotspot, all these questions should be answered after the vaccination. And this picture is from the first global pandemic of the information age, and it helps us better understand the value of the vaccination detection methods. And during different stages of the pandemic, as we all know that the emergent spreading epidemic stage, we need powerful tools for diagnosis and support diagnosis. And we need clear nucleic acid test and also serology test to help doctors assess the clinical purpose. But when the pandemic comes to the spreading or controlled stage after the academic stage, we need some other useful methods, especially when the vaccine are widely administered around the world. Neutralizing antibodies should be used to evaluate the vaccine-induced immune response of the vaccine receivers. And IgG, and total antibody with high specificity can be used to evaluate the herd immunity establishment after mass vaccination. So what is the difference between the neutralizing antibody and the IgM and IgG? First, let's review the structure of SARS-CoV-2. And as we all know that SARS-CoV-2 is responsible for COVID-19. And this virus contains 4 structural proteins: spike protein, usually we call it S protein, and nucleic capsid protein, called like M protein, amyloid protein, and membrane protein, and also the genome RNA. For IgM and IgG manufactured by Mindray, they are targeting the RBD on S protein and also the whole length of the N protein in order to enhance the sensitivity to achieve the aim of supporting diagnosis. But Mindray second generation COVID-19 products like neutralizing antibody is following another design. Spike protein consists of 2 domains, namely S1 subunit and S2 subunit, which are responsible for the binding step. S1 contains receptor binding domain protein, which is involved in the host receptor recognition and binding, whereas S2 contains the putative fusion peptide, as well as the peptide repeat HR1 and HR2. In humans, 3 types of antibodies or immunoglobulins have been the target of COVID-19 serology test. They are IgM, IgG, and IgA. The SARS-CoV-2 S protein and N protein are the primary viral antigens used in the current available antibody assays, such as Mindray first generation COVID-19 products. IgM and IgG are against the SRBD area and the N protein together. Just like I mentioned, the first generation is used for support diagnosis. Neutralizing antibody play an important role in viral clearance and have been considered as a key immune product by blocking viruses from entering host cells. Here, it is the schematic mechanism of the neutralizing antibodies. It is the competition of the neutralizing antibody with the receptor ACE for binding to the receptor binding domain, RBD, of the SARS-CoV-2 S protein. The protruding portion of RBD is both the ACE2 receptor binding site and the antibody's epitope. So this is the principle where the design of Mindray second generation COVID-19 products come from. But why we use neutralizing immunoassays instead of the gold standard virus neutralization test WNT? Usually we use the plaque reduction neutralization test, PRNT. As we all know, it is the gold standard to evaluate neutralizing antibodies produced by vaccines, but it requires handling live SARS-CoV-2 virus in BSL-3 laboratory. But it is also time consuming. It needs to culture cells, which typically takes 4 or 5 days. Because of these shortcomings, immunoassays are suitable and necessary to vaccine evaluation, both in clinical trials and after vaccination. So what is the intended use of neutralizing antibody measurement? The neutralizing antibody can be used to identify donors with high titers for convalescent plasma for the therapy of critical COVID patients, according to the guidelines recommended by FDA. And the second intended use is to evaluate the vaccine response or aid vaccine development. So what is the difference, the significance of the neutralizing antibody detection? Until now, over 40 vaccine projects are undergoing a clinical evaluation, but all these clinical verifications can only be carried out in a limited area and a few of countries, and cannot cover different populations. Also, there are individual differences in immunity, so chemiluminescence immunoassay can quickly and accurately indicate the immune status of the population after vaccination and help the governments judge the herd immunity of their countries. Besides the specific tests, there are also some important ordinary lab tests. Although the essential role of the clinical laboratories in this pandemic extends beyond etiological diagnosis of the COVID-19, biochemical monitoring of COVID-19 patients through in vitro diagnostic testing is crucial for assessing disease severity and progression, as well as monitoring the therapeutic intervention. Several common in vitro diagnostic tests have been implicated in unfavorable COVID-19 progression, potential providing important prognostic information. A recommended test list based on this current literature is included below, along with the major laboratory abnormalities associated with IDAPT COVID-19 patients and their potential clinical indication. In addition to more common laboratory tests, new evidence suggests that patients with severe COVID-19 could be the risk for a cytokine storm syndrome. Cytokine tests, especially interleukin-6, may be used where possible to assess severe patients suspected of hyperinflammation. Those specific and ordinary lab investigations are essential to the entire management of the COVID-19. Let's see what can Mindray provide. For the clear part, we have the first generation IgM and IgG ICs, and the second generation COVID-19 products, neutralizing antibody ICs. And as we all know that the first generation can support the diagnosis of COVID, and the second generation neutralizing antibodies can indicate the immune response induced by the nitro infection and vaccines. And for the sample type, the doctor can use serum and plasma. And it is easy to operate, and the time to first result is around 30 to 40 minutes. Besides, we also provide different models of instruments, and the throughput can reach up to 480 tests per hour, depending on different models used. And all these instruments are fully automatic, which can minimize the infection risk. And we can also provide different packages, such as 50 tests per bottle and 100 tests per bottle, which can satisfy different requirements from our customers. Here is the IC design of Mindray first generation COVID-19 products, IgM and IgG. And the IC design is ALP/AM/PPD system, and reaction mode is indirect method. Based on this design, Mindray ALP/AM/PPD reaction system, our R&D department designed this IC as a two-step, two-separation method. And this diagram shows the detailed reaction steps. There are some characteristics. The first one is using full-length recombinant N protein from E. coli and RBD fragment from the eukaryotic cells expression system to code the paramagnetic particles PMP to achieve high sensitivity and specificity. And the second one is use anti-human IgG to mitigate the specific IgG from the samples to impact IgM detection by competitive binding PMP, which is the potential risk for indirect format for high IgG samples. And the third one is using Mindray cocktail blocking formulation to minimize the non-specific binding and RF interference, which is also a potential risk for indirect format. And the last one, optimize the IC reagent formulation and the kinetics to achieve performance meeting with design requirement. Since the first generation product IgM and IgG were launched, we have done a lot of evaluations in global market. And until the end of 2020, we have collected more than 2,000 data from 19 hospitals and labs in 13 countries, 1,070 from positive samples and 1,017 from negative samples. And the sensitivity and specificity are listed here. We can see that from the PCR confirmed cases, the sensitivity of IgM and IgG together is around 98%. And for the giant, the specificity is around 95%. The performance seems good. We also did some evaluations in different countries and got some evaluation reports from different customer sites. And also the sensitivity and specificity are good and can satisfy different requirements of our customers. Our customers published some scientific papers, and all of them are listed here, show the good sensitivity and specificity of Mindray IgM and IgG. And these papers shown Mindray IgM and IgG ICs with chemiluminescence methodology had better traceability and more objectivity than rapid test. So clear test is highly recommended with testing on automated platforms and especially for a large scale of sample testing. These are the pictures of our evaluation sites all around the world. Almost all the evaluators were satisfied with our IgM and IgG. These are the customers' feedback from the evaluation sites. And from these pictures, we can see that all these evaluators are satisfied with the performance of the ICs. So in summary, Mindray SARS-CoV-2 IgM and IgG are fully verified and validated. No adverse events occurred. Most of the evaluations were excellent. The performance can definitely meet the requirements and the expectations of the customers. Here comes the second generation products, and the second generation we have 3 products, and one is neutralizing antibody core, and the second is neutralizing antibody RBD IgG, and the third one is neutralizing antibody RBD total. And so from the tables, we can see that the total sensitivities of these three products respectively are slightly different. And for the neutralizing antibody and neutralizing antibody RBD total has the highest sensitivity, and the other two are slightly lower than the neutralizing antibody RBD total. Because all these three assays have different designs for different clinical scenarios, there are three products. These three products may evaluate neutralizing antibody generation after nitro infection and vaccination. And Mindray is now cooperating with the Chinese vaccine company to evaluate the relationship between the neutralizing antibody tested by clear and also tested by PR and T using vaccinated samples. The experiment will be finished within 1 month. And as we all know that there are different types of vaccines in activated vaccines like Sinovac, RNA vaccines like Moderna and Pfizer and BioNTech, and also adenovirus vector vaccines such as AstraZeneca and Sputnik 5. So we still need more evaluation data of vaccinated people who receive different types of vaccines. So doctors and professors who are interested in this field and can also assess these kind of samples, please contact with me at this email address. I'm looking forward to your letters. This slide is about the discussion on SARS-CoV-2 and antibody types, functions, and possible clinical scenarios, and for the first generation IgM and IgG. And because both of these assays are targeting to RBD on S protein and N protein, so the possible clinical scenario is to aid the diagnosis and can indicate the disease courses, and these two assays have already been launched. And for the second generation neutralizing antibody core and RBD IgG and S total and RBD total, and they are following three different designs. So these three products may indicate the vaccine-induced immunity. Besides, for the RBD IgG, it can also indicate the past infection and can be used for prevalence screening. And the RBD total, can also use as an aid to diagnose COVID and can be used for prevalence screening also. These three products are still under development. And there are still some unknown questions left for COVID-19 and vaccines. And what is the dynamic changes of the neutralizing antibody, and how long do antibodies confer immunity, and from that one on is patient protected? So we don't know these up to now. So it is a summary. Identifying neutralizing antibody is important to know the immune response induced by the nitro infection and vaccines. Identify neutralizing antibody is also significant for vaccine receivers to know the real response before vaccinated. It makes sense for the government to reasonably distribute the limited vaccines to the populations in priority, like healthcare workers. In addition, Mindray also provides some other solutions for COVID-19 management, like biochemistry and hematology, and we provide 61 biochemistry parameters, and these are the parameters I listed here, are very useful to COVID-19 assessment, prognostication, and the therapeutic monitoring. For the hematology part, we can not only provide the reporter parameters, research parameters, but also can provide lab expert software platform, which is very, very useful and convenient for our customers. Okay, thank you for your attention. That's all.
Hello, everyone. I'm Chen from First Hospital of Zhejiang University, China. It's my great pleasure to be invited by Philippine Society of Pathologists and to be here to talk with you about our research progress on the laboratory diagnosis of SARS-CoV-2 infection. Today, the topic of my presentation is SARS-CoV-2 laboratory diagnosis, the experience and the problems. As we all know, laboratory diagnosis plays a decisive role in COVID-19 diagnosis. While many factors will affect the results of our laboratory tests. Among them, specimen type and collection time are the key factors affecting nucleic acid test results. Here is a paper we published in CCA recently. We enrolled 65 confirmed COVID-19 patients and collected different types of samples at the same time for each patient, including throat swab, nasal swab, and sputum. Although in the case of no sputum, we would collect a specific saliva after deep cough from them, also called Hawk luge saliva. As shown in this table, the pulse rate we tested was significantly different from different sample types. We can see that the pulse rate of stool swab was about 40%, which was relatively lower than those in other samples. For nasal swabs, the pulse rate was much higher than throat swab and was up to about 70%. We can also see from the table that 95% of sputum samples were test positive. So as the Hawk luge saliva samples, whose pulse rate was up to 88%. And not only is the difference of the pulse rate for the nucleic acid test, we also found the different concentrations of nucleic acid among different types of samples. We can see that in stool swabs, the concentration of nucleic acid is relatively low, which explains why many samples in the stool swabs are weakly positive. In contrast, nucleic acid concentration in nasal swabs, sputum, and saliva were relatively high. In addition to samples from the respiratory tract, positive virus nucleic acid were also detected in stool, blood, urine, and corneal secretion. Here is another paper we published in British Medical Journal. We can see that positive viral RNA are detected in stool from 69% patients, from 59% patients, and 41% patients were tested positive for viral nucleic acid serum samples. Moreover, given that the amount of nucleic acid in stool samples is high, we isolated the virus from the stool successfully. However, although we detected viral nucleic acid in blood, urine, and corneal secretion, we did not culture the virus from these three samples. We not only culture the virus in stool, but also successfully infect human intestinal epithelial cells using the isolated virus. So the fecal to oral transmission route is clear. According to our further study, our dynamic monitoring found that joint detection of different specimen types is beneficial to improve the positive rate. We can see from this figure that the blue lines represent nucleic acid test results in the stool, and the virus nucleic acid positive time in stool is very long. Moreover, in mild cases, the viral nucleic acid in the stool was still positive, while the nucleic acid in the respiratory specimen is negative. Therefore, we have included this finding into COVID-19 treatment guidelines, in which we require patients to be tested for viral nucleic acid in stool before discharge from hospital, even if the virus nucleic acid is negative from respiratory tract samples, so as to ensure 2 consecutive negative fecal viral nucleic acid, which will be safer for patients' discharges. We can also see that the viral nucleic acid level or dynamic monitoring of load is associated with prognosis. In this figure, red lines meant the viral nucleic acid level of severe patients, which was much higher than those of mild patients. Additionally, the peak of virus release is about a week after the onset, and it peaks about 2 weeks after the onset. So dynamic monitoring is something that can be very reliable. Here is another interesting paper published in the New England Journal of Medicine recently. In this study, the researchers cultured SARS-CoV-2 in cereal nasopharyngeal or oropharyngeal samples obtained from hospitalized patients with COVID-19 to assess the duration of shedding of viable virus. We can see from this figure that the red dots represent samples positive for virus culture, and the blue ones are negative. This research found that if CT value is more than 28, the results of wireless culture will tend to be negative. They also found that with longer disease progresses, it becomes more difficult to get possible wireless culture, and longer than 12 days after the onset, all the results will be negative. Thus, for improving virus culture, it's better to get samples from the lower respiratory tract of the patients who are at late stage. According to our results, we suggest the cautions in specimens collection. First, if the patients have sputum, it should be collected first. In the case of no sputum, we recommend saliva after deep coughing. We also recommend that during outpatient sputum collection, we can allow patients to collect the sputum or saliva after deep coughing without removing a mask. In this way, suitable specimen can be collected and the environment can be protected to avoid cross-infection. Second, improving the quality of nasal swab and slot swabs. To this end, we can use deep sputum aspiration or alveolar lavage fluid for severe hospitalized patients. Since the postulate of this lower respiratory tract specimen is very high. Third, we can use blood, stool, and other suspicious specimen when necessary. Next, let's move forward to this slide. This slide shows a typical CT scan of a COVID-19 patient. We can see that the virus infection happened in the pulmonary interstitium, which is the outer margin of the lung, far away from the airway, explaining why patients would have a dry cough. Although this wise amount of virus discharge in each sample we collect varies greatly, and there may be inconsistency in the continuous sample test. Considering that the target cell of SARS-CoV-2 are mainly located in the lungs and the slot residence time is short, slot swab can appear negative, especially in later stage. The vast majority of the nucleic acid level in slot swab is very low, often undetected. The sputum specimen of lower respiratory tract postulate is very high, which is suitable for the monitoring of critically ill patients in the late. In addition to the factors before analysis, the quality control of a nucleic acid testing method and verification is also very important. Pre-treatment after specimen collection can have a significant impact on the results, especially for sticky specimens like sputum or saliva after deep coughing. We did a report in 28 on influenza A, and we diluted sputum in 3 different ways using saline, DDT, and protease K. As shown in this figure, we can see that the postulate of the samples diluted with DDT and protease K was significantly higher than that of the sample diluted with saline. And not only the postulate detected, but also the nucleic acid concentration obtained by these 2 methods was significantly increased. The graphic on the far right indicated that after the protease K treatment, the nucleic acid purity is very good. That means the nucleic acid quality is very good. In addition, the quality of a nucleic acid extraction also has a significant impact on the overall detection effect. Listed PCR kits tend to recommend some of their extraction methods. We have to evaluate the results found that PCR kit manufacturer recommend method is not necessarily the best. For example, extraction methods recommended by PCR manufacturers C and D were no better than B, so laboratory testing method is necessary to put into use before must evaluate the nucleic acid extraction system. Currently, there are lots of real-time fluorescence quantitative PCR kits, which are used very well worldwide, but there are still some differences. The 6 PCR reagents evaluated in our study had different detection sensitivity, and the 4 reagents A, B, C, and D are generally not bad, especially A and B, which has very good sensitivity. However, the sensitivities of E and F kits are not good. For PCR reagents, we mainly focused on the minimum detection limits and repeatability. The lower of the minimum detection limits, the better. We also hope that the CV of weak positive specimens can be controlled less than 5%. One of our experience is to use a limited number of specimens, gradient dilution, used to verify the minimum detection limit. For example, using a B kit, the possible results can be detected after serial dilution of the samples, but for D and F kits, the samples are undetectable after a certain dilution. Thus, the detective sensitivity of B kits is better than D or F kit. As we all know, real-time fluorescence quantitative PCR has 2 disadvantages. One of relatively slow, which generally takes 4 hours, and the other is very complicated, which requires special protection. We look at some new rapid or POCT molecular diagnosis methods used internationally. These methods are based on qRT-PCR or isosomal amplification. Then we also made a brief evaluation to compare with a classical real-time fluorescence quantitative PCR. Both of RAA technology of algorithm amplification technology and digital PCR have good sensitivity. Of course, these fast methods need to be fully verified when laboratories use them. When we worry about is that weakly positive samples will be tested negative after fast detection, though it is a development trend. Next, I will talk about some problems on serum antibody detection, which is also a great concern to us all. In our research that published in European Respiratory Journal, we conducted a follow-up analysis of 80 confirmed patients and found that with the recovery of patients, the viral RNA levels in the samples of the patients was decreasing. So about 7 days after the onset of the disease, we could measure the total antibody, which could include IgG, IgM, and IgA. We can detect IgM within 10 days of onset, which is the purple line in the figure, and IgG does not appear until 12 days after onset. This graph shows that antibody detection lacks the value of earlier diagnosis compared to the nucleic acid, and its significance may lie in epidemiological investigation. We have also observed some patients are still positive for IgM 1 or 2 months after they are discharged from hospital. Therefore, IgM may lack the significance of an acute phase indicator. Now we focused on methodological issues. We know that IgM is a capture method. We code it as antibody capturing all IgM antibodies in serum. The specific for SARS-CoV-2 IgM depend on the viral antigen we used. The method for measuring IgG is an indirect ELISA method with coded SARS-CoV-2 specific antigen. The second antibody used is not specific. For the total antibody, the method is double antigen sandwich method. In general, this method, such as the specificity of capture and indirect ELISA, is less than that of double antigen sandwich method, particularly affected by some specimen factors, such as rheumatoid disease or other reasons. it can lead to false positive. Therefore, the results of the antibody should be interpreted carefully, and we will repeat the test with the same or other reagents if necessary. Sometimes we found that dilution of samples is beneficial to eliminate the interference. Additionally, we should be very careful about interpretation of the positive specimens. Our test protocol is that antibody positive results must be reviewed, especially the colloidal gold rapid method. It produces false positive probability is relatively high because it's a very simple operation. Reaction time is very short. It's difficult to exclude contamination factors. As I mentioned earlier, IgM can be detected long after convalescent patients have been discharged from the hospital. We should be really careful about Ig positive antibody results. Not only exclude the false positive results to reduce our unnecessary prevention and control costs, but also confirm its positive results to ensure that we choose an asymptomatic carrier. So we must combine clinical evidence, epidemiological evidence, and even the results of the nucleic acid to judge the results of antibody tests. Currently, some diagnostic reagents for neutralizing antibodies in COVID-19 patients have been developed. We also investigated the neutralizing antibody response to SARS-CoV-2 after vaccination and infection using an automatic analyzer system. As shown in the left figure, the titer of neutralizing antibody to SARS-CoV-2 in patients were declined. We also involved 20 volunteers and tested neutralizing antibodies response after vaccination. We found that 15 days after second dose, which was shown in the right figure, the level of the neutralizing antibody was increased in serum samples. Next, other important indicators monitoring the treatment of COVID-19 in our laboratory found some significant blood routine results in COVID-19 patients. In particular, it was found that the majority of patients with COVID-19 showed a significant lymphocyte decline at the initial stage, and the progressive reduction of lymphocytes was closely related to the severe disease. We now know that the ratio of the neutrophil nucleic to lymphocytes can be a factor associated with severe illness in COVID-19 patients. We found that a significant increase in D-dimer in COVID-19 patients is often a predictor of poor prognosis. It was reported that many patients have elevated cardiac markers, mainly creatinine kinase and LDH. Abnormal liver function with ALT and RST elevating in hospitalized patients. In our opinions, due to the lack of evidence of direct viral damage to the liver, the abnormal liver function of COVID-19 patients may mainly be caused by antivirus drugs such as liver damage caused by Albendazole. Therefore, liver function monitoring is also of great significance for treatment. Severe COVID-19 patients, especially the elderly, also develop a series of renal insufficiency with proteinuria occurring in 60% and significantly elevated creatinine levels, particularly in patients prior to death. CRP is a very commonly used emergency indicator of inflammation, and we found that its significant increase in COVID-19 is also an indicator of poor prognosis. And of course, there are also changes in lymphocyte subsets, both CD4 and CD8 T cells, that are significantly reduced in critical ill patients. Since severe inflammation is related to cytokines, we also observed significant cytokine changes in severe patients, with many cytokines increasing and some decreasing. In particular, IL-6 is increased in 76% of severe patients. So this indicator can be used as an important indicator of inflammation monitoring. Recently, a research group focused on the routine laboratory testing results of patients and developed a machine learning model for the diagnosis and the prediction of COVID-19 using artificial intelligence. In this model, patients brought routine results and indicators of the coagulation functions, liver and kidney function were involved to diagnose and predict an individual's SARS-CoV-2 infection status. The performance of 4 machine learning models from 5-fold cross-validation are summarized in this figure, including GBTT, random forest, Rogers regression, and decision tree. Finally, they found that the GBDT model achieves the best performance with an area under the reservoir operation characteristic curve, a value of 0.85, sensitivity of 0.67, and specificity of 0.8. It was found that decreased white blood cells, lymphocytes, and calcium, as well as increased red blood cells and LDH, were more likely to predict a positive result of SARS-CoV-2 infection. In this study, among the 32 patients which SARS-CoV-2 RT-PCR results were initially negative, but upon repeated testing within 2 days were positive. This approach predicted positive of initial RT-PCR for 21 patients. Its sensitivity is 66%. At last, I want to talk to you about the detection of the pooled samples in a large population. There are currently 2 methods. One is to take 200 microliter of each specimen preservation solution, such as 5 samples after collection, and mix them thoroughly for testing. The second is to collect slabs, such as 5 or 10, will be immersed in the same specimen preservation solution before testing. The advantages of both approaches are cost savings and time savings, and they are stable for screening population in low-risk areas. However, it has limitations because it is a mixture of multiple specimens, and there is a risk of missing the test due to dilution or interference with multiple swab proteins. So we emphasized that the PCR reagent used for this test is highly sensitive PCR reagents. Even so, we recommend that mixed testing is not appropriate for screening in high-risk area, much less for patients in hospital with suspicious populations. Above are some of our appeals and suggestions on the SARS-CoV-2 testing and diagnosis. Very happy to take your question. Thank you for your attention and listening.